Thank you, Maggie, for that introduction, and happy to be here talking to you about genetic solutions that we bring for your immuno-oncology research. So let's begin by taking a look at what is immuno-oncology, a very brief introduction. So I'm going to skip through the slide and move on to immuno-oncology. Immuno-oncology is that branch of cancer therapy which uses the body's own immune system to trigger a response against tumor cells and ultimately eliminate them. In the more recent past, immunotherapy has shown to be extremely effective in providing a response and sometimes complete remission for some patients with advanced cancers, and especially those which had patients who had not shown response to treatment, such as chemotherapy or radiotherapy. Because of this, immuno-oncology or immunotherapy is considered the fifth pillar in cancer therapy, along with radiotherapy, surgery, chemotherapy, and targeted treatments. What's also notable is immunotherapy has found to be actually pretty successful in treating advanced stage cancers when used in combination with traditional therapies such as surgery, chemotherapy, and radiotherapy. So what makes immunotherapy so different? Well, I think it can be best described because immunotherapy or immuno-oncology uses the adaptive immune system to recognize and specifically attack cancer cells. The entire process by which this happens can be easily described in the cancer immunity cycle, which was proposed by Melman and Chan and can be seen in this figure here. These are a series of stepwise events that must be initiated and allowed to proceed iteratively for an anti-cancer immune response to be effective. T cells and T cell mediated responses are the focal point of any immuno-oncology research. So that's a very brief overview of what immuno-oncology is. And currently, the immuno-oncology focus areas can be broadly classified into 6 main groups: the checkpoint inhibitors, adaptive T cell transfers, or cell therapy, more popularly known as therapeutic cancer vaccines, oncotic vaccine immunotherapy, monoclonal antibodies, and cytokines. For the purpose of this talk, we will focus on the first 3 focus areas. That's the checkpoint inhibitors, CAR T cell therapy, and therapeutic cancer vaccine. So let's take a look at what checkpoint inhibitors are. So checkpoint inhibitors are essentially proteins that are present on the surface of tumor cells, which help the tumor cells to evade body's immune system. And how do they do that? They do that by essentially fooling the immune system into thinking that the tumor cells or cancer cells are self instead of foreign. And checkpoint inhibitors, on the other hand, are molecules that activate the immune system into recognizing tumor cells as foreign and thereby causing them to eliminate the tumor cells. Now, targeted inhibition of checkpoint inhibitors, targeted inhibition of checkpoint receptors of proteins can enhance the response of T cells towards the tumor, and this is what is used in checkpoint therapy, immune checkpoint therapy. And currently, immune checkpoint inhibitors are considered very, very important and probably the most studied targets in immuno-oncology. Cancer vaccines, like most of the vaccines, therapeutic cancer vaccines trigger the immune system into recognizing and attacking markers or antigens that are present either in or around cancer cells. multiple types of therapeutic cancer vaccines could be, either proteins, whole cells, or antigen-presenting cells, typically the dendritic cells. And cancer vaccines often do require additional substances such as adjuvants for optimal effectiveness. And finally, CAR T-cell therapy. So what is CAR T-cell therapy? It's a very interesting molecular mechanism in which the T-cells from a body are removed. They're genetically programmed to fight cancer, multiplied, and then infused back into the body. These newly programmed T-cells, or CAR T-cells, are now able to eliminate tumor cells. CAR T-cells have been successfully engineered to treat acute lymphoblastic leukemia when no longer responds to treatments. And in the past couple of years, 2017-2018, FDA approved 2 CAR T-cell therapies for B-cell lymphoma and acute lymphoblastic leukemia. And more research is being done to further find applications of CAR T-cell, but it's extremely promising. So with that, I will shift gears and talk a little bit about what tools we have in our genetic toolbox from Applied Biosystems and Ion Torrent, which could be used in your immuno-oncology research. So here is an overview of all the different platforms we have within our genetic analysis toolkit. So we have solutions based on the electrophoresis or CE. We have solutions for PCR and digital PCR, microarrays, as well as NGS. So essentially, the whole plethora of genetic analysis solutions. So in the next few slides, we'll go take a deeper dive into which of these solutions can be used for which of the immuno-oncology research and some examples of how in the past customers have used them. All right. So first up is checkpoint inhibitors. And for checkpoint inhibitor research, we have solutions which are in the microarray platform, qPCR, as well as NGS. So I'll spend a little less time on this and subsequent ones because more examples and it will help you understand how these can be used. So for microarray, we have our whole transcriptome analysis panels as well as CNV assays. In qPCR, we have a multitude of TaqMan gene expression assays, which are present as different panels, pre-selected panels, and they can also be ordered as custom gene expression panels based on your custom requirements. And of course, we have the NGS solution where we have whole transcriptome panels, both human and mouse, as well as more targeted panels such as the Oncomine Immune Response Research Assay or the TCR beta, SR, and LR assays. All right, so let's take a look at where some of these solutions have been used. So first up is a study we did internally, which I'm going to talk about. And more details of the study can be found in an upcoming webinar by my colleague Steve Jackson, where he's going to talk about how TaqMan gene expression assays can be used to study any gene signature in a tumor microenvironment. So why is this important? We found that there are a lot of applications in which researchers are looking at genes of a tumor microenvironment because they create a distinct molecular profile, and the TaqMan gene expression assays could be a great choice to look at these genes in the tumor microenvironment. So don't forget to tune in for that webinar, which is coming up. And on the right, you're looking at data from a publication in Cell from 2019, where the authors used TACMAN gene expression assays to characterize expression of various immune-related genes in dendritic and macrophages cells in response to BMP4. And they found that this information was critical in their ability to characterize the dendritic and macrophages differentiation in acute lymphoblastic leukemia. So that's an example of where TACMAN gene expression assays can be used. Next up is microarrays. So as I mentioned, microarrays, we have a couple of different panels. The most useful in immune checkpoint inhibition research are the Clarium DB assays, which are suitable for deep transcriptome analysis, coding and non-coding RNA, as well as alternative splicing events. We also have Clarium SBQ assays, which are suited for gene-level expression profiling of well-annotated genes. And both the Clarium D and Clarium S assays have very high sensitivity, and the entire transcriptome can be characterized with as little as 100 picograms of RNA. All right. So coming to an example or a case study, we looked at a publication in Frontiers of Pharmacology, again in 2019, where the entire transcriptome was characterized in response to the effect of sphingosine kinase in metastatic prostate breast cancer cells. And at the time of the study, sphingosine kinase, both SK1 and SK2, were pretty well characterized proteins. And there was a lot of information already available as to which pathways they belong to and which pathways were up or down regulated in response to these particular proteins. However, this study revealed that many pathways that cross-linked with sphingosine kinases were either up or down regulated. So pathways such as RAS, MAPK, GTPase, PIK3 were upregulated in response to SK knockdown, whereas several other pathways which were traditionally known to be associated with cancer progression and apoptosis, such as PL2 and PLP, were not affected by SK knockdown. So this kind of analysis, where there's a lot of interaction of pathways, especially in tumor progression, it's critical to be able to look at the entire genome or the entire transcriptome while making decisions on which pathways may be affected in response to a particular knockout. And Clarium assays can be a good tool for this analysis. All right, so moving on to NGS. So with NGS, we have, of course, the whole transcriptome analysis, but we also have many targeted sequences, targeted sequencing panels. And one of them is the Ion Torrent Oncomine Immune Response Research Assay. This is an assay which consists of 395 genes, and they have been custom selected because these genes are known to be, involved with the immune response, the tumor immune system interactions, right? So all 395. And what you're looking at here in the graph is expression levels of a subset of these 395 genes across 10 pan-cancer samples. And the blue box plots refer to genes that belong to the Oncomine Immune Response Research Assay. And you can see a pretty well spread out dynamic range of the genes, as well as these are highly sensitive. And the gray bars are is data for the same sample from a competitor platform. So again, a nice tool for a targeted sequencing approach. All right, so within our NGS toolkit, we have some other specific targeted sequencing panels. One of them is the Oncomine TCR beta LR assay. Now, this assay is focused and enables TCR sequencing, which is being considered as a possible biomarker for future immunotherapy responses. And in a study that was published in 2019 in Frontiers of Immuno-oncology, the authors used iron sequencing technology with so the TCR beta assay and found that they were able to sequence and accurately assess TCR coverage because with the Oncomine LR assay. And they especially noted that TCR convergence may be masked in other sequencing technologies, and hence the choice of sequencing platform for this kind of research was very important. Right? So these are some of the examples that we had for how our tools can be used for checkpoint inhibition research. We will now shift gears to talk about therapeutic cancer vaccines. So therapeutic cancer vaccines also can use a lot of tools that were meant for immune checkpoint inhibition research, but in addition, we also offer OncoScan CNV assays and the Cytoscan HD suite within the Microarray platform. A lot of the TACMAN assay panels can be used for therapeutic cancer vaccine, of course, and a lot of the panels that I already mentioned in NGS, including the Oncomine TCR beta assays, ion ampliseq transcriptome, the exome ion ampliseq exome kit, can be used for cancer vaccines. And of course, almost all of these platforms also offer custom panels which can be used for, to create your own custom assays within a panel and study. Right, so let's look at some exciting work that was done in the field of therapeutic cancer vaccine using our tools. So the heat map that you see to the left is from a publication in Frontiers in Oncology from 2019, where the authors used applied biosystem Clarium S mouse assay to analyze the whole transcriptome in neoplastic tissues, and this analysis led to the identification of 7 antigens which were highly expressed in tumor cells but not in normal cells. These 7 antigens were then used as targets for vaccine development. In a similar study, the authors of a publication from 2018 in Journal of Immunotherapy used tactic expression assays to actually study the effect of a DNA vaccine, the MIP3 alpha GP100 DNA vaccine. Effect of it was used to study, and they found that efficiency of this vaccine, and varies for the cancer vaccines, was dependent on tumorigenic factors in the tumor microenvironment. So again, an example of how very targeted tools can be used for such analysis. All right, and then we have an example where the ion torrent Oncomine TCR beta assay, along with the ion torrent Oncomine immune response research assay, was used to evaluate TCR convergence, and this TCR convergence was used as a potential biomarker for response to dendritic or peptide-based vaccines in advanced-stage melanoma. Pretty neat. All right, so those were the examples for therapeutic cancer vaccine. And next up is CAR T-cell therapy. So just like previously, we have solutions in both qPCR and NGS, but I do want to point out that we also have CE-based solutions for CAR T-cell therapy, and there's a very neat study that was published recently, where the Applied Biosystems genetic analyzer, 3500 genetic analyzer, was used to characterize CD45 RA positive or negative, and this was done because CD45 RA is being investigated as a potential biomarker for leukemic stem cells. So a neat platform to characterize that molecule. And in some of the other studies, we have Clarium S assays, which were used for whole transcriptome analysis, and to characterize, CAR T cells, and these cells were characterized using Clarium S assays both before transplantation. So when these cells are generated in vitro, they are characterized, as well as their progeny after in vivo maturation. And the results give important insights into differentiation of the progenitor cells, and are critical in making decisions about success of genetic engineering of CAR T cells. And finally, Taqma gene expression assays have been used successfully to study the validity of B-cell maturation. So BCMA is considered a novel CAR T-cell marker, and gene expression assays such as Taqma gene expression assays could be very useful tools in studying the validity of such antigens. All right, so with that, I will come to my summary slide. And this is just, again, a brief overview of the different platforms we covered in the course of this presentation. So we looked at how whole transcriptome microarrays such as CAR-T and S assays can be used for whole transcriptome analysis, depending on specific applications. That we've seen, examples of how these were used for checkpoint inhibition and vaccine, as well as characterization of CAR T cells. qPCR assays, of course, we have a plethora of qPCR assays which are very well characterized, and they can be useful tools for discovery and verification of checkpoint inhibitors and other immune-related genes, such as cytokines and chemokines. So within the Sanger sequencing and fragment analysis by capillary electrophoresis platform, we have the Seq Studio genetic analyzer, and this can be very useful for TCR sequencing and for analysis of any sequences up to 700 base pairs. And coming to NGS, of course, we have a lot of assays which can be used for immune checkpoint, for immuno-oncology research in general. We have the more targeted sequences are the Oncomine immune response research assay and the Oncomine TCR beta LR assays. So with that, I come to the end of this presentation. And I'm happy to take any questions that might be there on the call.